Good evening to all of you. I am Dr. Sanjeev Pandey. I'm an associate professor in the Department of Botany at Banwarilal Bhalotia College, Asansol in West Bengal. I'm going to make a series of lectures about recombinant DNA technology for the students of BSc honors and also program of botany and other streams of life sciences, including biotechnology, because this part is common to all of them. So let us start with definition of recombinant DNA technology or genetic engineering or gene manipulation. These are the alternative terminologies used to define the same thing. So basically it's the techniques that are associated with the construction and application of this recombinant DNA is referred to as this genetic engineering or gene manipulation. So what is a recombinant DNA? A recombinant DNA is a chimeric DNA. That is, it's a mixture of two different DNA generated in vitro by covalently joining DNA molecules from different sources, like this one. This is a chimeric DNA or recombinant DNA. The orange part is the gene of interest that has been obtained from some other organism, and the black region is a vector part. It is a plasmid vector within which that gene of interest has been inserted. So what it makes is a combination of two different DNA, the plasmid DNA and the gene of interest. So this is a recombinant DNA or chimeric DNA, you can say. So to make a recombinant DNA like this, there are 5 things that are required to be done. The first thing is you need a vector. You need a vector DNA within which you can insert your gene of interest. Secondly, you also need the gene of interest. You also need to know what gene you want to clone and where from you want to obtain it. We call it the foreign DNA or the insert DNA. Thirdly, you require several different enzymes. The most important among them, if you narrow down, the most important among them are the restriction endonucleases, by which you introduce cuts in the DNA, and ligase, with the help of which you join the DNAs. And there are several modifying enzymes with the help of which you make your cloning successful, like the shrimp alkaline phosphatase, the T4 polynucleotide kinase, the DNA polymerase 1, clenofragment, the exonucleases, the 5' and 3' exonucleases, the terminal transferase, and many other like Taq polymerase, and maybe the reverse transcriptase sometimes. So, and you require an instrument by which you can monitor these changes that are taking place. So you require electrophoretic apparatus, particularly the agarose gel electrophoresis. You need to perform that to understand that your cloning is successful or not. And you also need a host cell within which you will transfer the recombinant DNA so that it can remain there and it can replicate. It can make several clones of it. When the cell divides, the DNA also replicates and multiplies, and the number increases, it amplifies, and anytime you can purify and you can go ahead for further research, whatever you want to do with that. So these things are required. The host cells could be E. coli or any other system like eukaryotic system also, like yeasts, but mostly it is E. coli, within which the first part of the recombinant DNA technology is performed. After that, there are several other things that can be done. Not the only objective of recombinant DNA technology that you will try to make a genetically modified organism. There are many other objectives or motto of genetic engineering.